Okay, so now we're going to talk about another approach that people have taken to this same question, which is, sure, you found that you could distinguish two things, but you want to know why. You want to know what the basis is on which you could distinguish them, or what information the population represents that you're studying. So we started the binary classification. Just say, note that everything I told you about here is all binary, right? All the way through, you're always testing one thing versus another thing. It's always 50% chance. So as you do these different levels of generalization, you can test what the basis of that binary classification is, but always by doing a new binary classification, right? Is it one thing or the other? Is that clear? Okay. So here, right, we have two dot clouds. And the binary question is, can we distinguish them? Is purple different from green? But you could imagine asking a question, which is, what's blue more similar to, green or purple? And obviously, that's just a very small generalization of the idea of asking, is purple different from green, right? You just put one more sample in there and then ask, which one is it more similar to? But this kind of expands the questions that we can ask, because now we have a measure of the, or could have a measure of three things, right? How different purple is from green, how different green is from blue, and how different blue is from purple. And so once you have your data in this kind of, once you have data like this, right, at least three conditions, then we can take these data and think of them this way. The question we were asking before is, if you get a new instance of the same condition, right, a new instance of female happy, is it more similar to your old examples of female happy than it is to your old examples of female happy? That's a way of thinking about the classification question. Is that clear? So another way of thinking about what the classification question is when I get a new example of a blue, is it more similar to my old examples of blue than it is to my old examples of green or purple? Right? And so the classification question in this form is asking, you can think of this as the training examples, and this is the testing examples, right? So if I have, these are my training data clouds, these are my testing examples, are the ones that match more similar than the ones that don't match? In the three condition example, this is a description of the hypothesis that you can classify those three conditions. That for each condition, a new example is more similar to the training set examples from the same condition than to the other two. Yes? Okay. But what's cool about thinking about the data this way is that in addition to showing you that you can classify, I just drew this as a qualitative picture. Todd saying, what actual kinds of numbers would you put in there? Right? We could stop here and think, what kinds of numbers would we put in here? Right? If we're doing it the way we've been doing it before, right, then this could be classification accuracy. Another option is that it can be split half, right? So if I had one example of blue in my training set and one example of blue in my testing set, we can just ask, is the distance, actually it doesn't have to be split half, it could be any way that you split your data, so let's just call it distance. So if we say, here's my blue cloud, now I have a new example of blue, right? Is the distance of the new example of blue to the training examples of blue less the distance of the new example of blue, training examples? There's many different ways to measure distance, as we talked about, but in a space, one simple way is Euclidean distance, right? So this could be just Euclidean distance. For this purpose, we don't have to do any clustering, right? So we could literally just do what I've done, that is, take some data, learn a cloud for the blue, right? Learn the distribution of the blue data. So here's a very simple thing you could do in the training, right? So the training tells you all of these are examples of blue. All of these are examples of green. Now you have a new instance, right? Obviously, there's many different metrics you could use for distance, but a very simple one is take the mean of each of your samples and calculate Euclidean distance of the new instance to each of those. That's not the only way to do a distance calculation, but exactly which number you put in here is sort of a mathematical question, but the conceptual question of what are you trying to ask doesn't depend on which exact number you put or which exact metric of distance you use. Are people following the difference I'm trying to articulate? So there's one question which is actually mathematically how do you calculate the distance of the new instance to the training? And that there are options. But another question is why are you calculating the distance of the test data to the training? That's as an implementation of this more generalized idea of whether in the pattern of activation you can classify the stimulus. People following at least the qualitative question, if not the quantitative one? Okay, so the reason why I'm showing it to you this way is because this makes apparent that once you have 3 conditions, if you only have two conditions, classification and representational similarity analyses are exactly the same question. Right here, I'll draw you the 2-condition version. If you have 2 conditions, the question, can you classify blue from green, is the question, are the diagonals greater than the off diagonals in this matrix? Right. So what we were asking here is we had trained on some examples of female sad, some examples of female happy. We got new examples of female sad and new examples of female happy. The question, can you classify the new examples based on what you learned from the examples, is the same question as, are the new examples more similar to the old examples of the same condition than they are to the old examples of the other condition? Right? Okay. And so the diagonal is the new examples of the conditions being matched to their matching old example. The off diagonal, this one, is for each new example, how similar it is to the old example of the other condition. And when the diagonals are higher than the off diagonals, then you can classify numerically. Right? You have to repeat it to people to find out if you can statistically. Numerically, what classification is that the diagonals are higher than the off diagonals. Yeah, so this representation summarizes across all the iterations. Right, okay. Generally, yeah. So I could calculate, every time I leave some data out, I can calculate the distance of the new example to the old example. But what I'm depicting here is then averaged across all those iterations. Right. What's the average distance of the new example to the old examples? Right. But yeah, so it helps to start here, which is to say, if the new example of blue is pretty similar to the old examples of blue on average across all iterations, and the new example of green is pretty similar to the old examples of green across all iterations, that's this one, and the new greens are not very similar to the old blues, that's this one, and the new blues are not very similar to the old greens, that means that there's a pattern difference that lets us classify blue from green. You couldn't test this as an interaction because none of the cells are independent of one another. Right? Because remember, when you classified green with this new example, you asked which one is it more similar to, or what's the distance to each one. And because each of these is a pairwise relationship, they're not independent cells. So yeah, this is one thing that's actually funny about how to think about the data in a similarity matrix, is that the cells are not independent of one another. And if you know all but one, you can figure out the last one, for example. Yeah. Okay, but so again, what I'm trying to show you is that this representation is a different representation of the same question we were asking before, right? We asked a binary question about classification. This is a representation of that same binary question, right? This is also a representation of the same binary question. We have a third condition, but we're still just asking, for each condition, is it more similar to itself than it is to anything else? So far, right? But what this shows, once you do it this way, is that there's not only information available about whether each condition's new example is more similar to itself than to anything else, but also here, in the off diagonal, is information about how similar the conditions are to each other, right? So you could have blue is more similar to itself than it is to anything else, green is more similar to itself than it is to anything else, purple is more similar to itself than it is to anything else. That's one piece of information. But now we also have information about how similar is purple to green, how similar is blue to green. And those two numbers don't have to be the same, right? Purple could be more similar to green than blue is to green. So whereas in the binary case, there's nothing in the off-diagonal particularly. Once we have more than two conditions, the structure in these other cells could get interesting, right? Could tell us something about the similarities between the conditions to each other, not only the separation of each condition from the other. So these are all binary pairs, right? So you can think of it as a classifier, or in this case, again, you can think of it as a distance, the way we drew it here, right? So I have one new data point, right? This one blue square, and I can ask, how far is it from the old blue cluster? That's this number. How far is it from the old green cluster? That's this number. How far is it from the old purple cluster? That's this number. And then for one new green point, you would do it each time. When I showed you this experiment, I didn't ever ask, how similar are social cartoons to male happy? How similar is male happy to female sad? I didn't compute all the similarities. I could have. Right. But the logic of this analysis is slightly different from the logic of this analysis. In this case, in the classification and generalization, we learn one distinction and ask what it can be applied to. And that's a way of saying, for this pattern that I learned, what dimension is it classifying? I'm going to show you what the kinds of things you can do with this, but it's a slightly more general set of things, right? So as you say, we could generalize the question here into a representational similarity matrix and ask about all the different similarities. But so far what I did is I took one pattern and then asked what was the basis of that pattern. So I'll come back to how you would draw this test in representational similarity matrix. You can certainly implement this generalization test in a representational similarity analysis, for sure. Okay. Yeah. Okay, representational similarity analyses in general are a more general formulation. This is not, it depends exactly how you implement it, but in principle you can encompass everything you would do in a classification and generalization analysis. You can encompass that and more in a representational similarity analysis. Yeah, so it's in some ways a more general form. Before we had a question, is each condition more similar to itself than it is to anything else? Now we have the question, is each condition more similar to itself than it is to anything else? But also, what are the relationships among the conditions with each other? So that even when you're not a match, you could still be closer or further, right? So this blue point is not a green point or a purple point, but it is more close to the green ones than the purple ones. Okay, and so this way of thinking about multivoxel pattern analyses lets us think about any theory that predicts how similar two stimuli are. Not only that it's another instance of the same thing, but actually how similar it is to anything else. So the huge benefit of representational similarity analyses is that this format of data, how similar one thing is to another thing, you can derive it from anywhere, right? So here's the first thing you can do. Okay. So one thing we could say is, a priori, I have two different theories about these data that I'm interested in testing. And one theory predicts that green and purple are similar and blue is different. And another theory predicts that blue and green are similar and purple is different. So one way to think about this is, say blue and purple share shape and green and purple share motion. And this says, I think that motion is what matters. And this says, I think that color is what matters, shape is what matters. I'll draw this. So if I did an experimental design where I manipulated two different features of the stimuli, shape and motion, right? And I want to know for this brain region, which feature does it care about? Does it care about the shape or does it care about the motion, right? Then this is my prediction for a region that cares about the shape, right? That it would treat the two as more similar that have the same shape, and it would ignore the motion. And this is my prediction for a brain region that cares about the motion. It would treat the two as similar that share their motion and ignore the similarity. Is that clear? Okay. So this then, do one experiment, collect data on the pattern of response to each of my three conditions, say, for example, in MT, motion brain, right? And then once I've collected the data for these three conditions, again, using some kind of distance measure, I can ask how similar each condition is, both to itself and to the other two conditions. You can see that these two predictions, they both predict classification, right? Whether you care about shape or motion, you should be able to classify the stimulus, right? The diagonal is higher than the off-diagonal in both predictions. What's different is in the off-diagonals, right? Which of the off-diagonals is higher? And so now a way of asking, sure, we can classify these stimuli, but is it because of their shape or is it because of their motion? Is to ask which of these two predictions is a better fit to my data. Is that clear? And so now I say, if this is a better fit, then it's motion. If this is a better fit, then it's shape. And at this point, these are two matrices, and you can just do a correlation between them, a Pearson's R, for example, right? And you can ask, is this is the motion R value, shape R value, and we can just ask, is the motion R value higher than the shape R value for MT across subjects? So this is a way of testing which of two theories is a better theory of the information in the patterns in a brain region. And what's cool is that we related data we collected in the brain to a theoretical prediction by putting our theoretical prediction in the format of a representational similarity matrix, and then we can just ask about the fit between that and the data that we measured. This is a very simple case, so I'm going to work to more complicated cases, but it helps to see the simple case before we start going to more complicated cases. The point that Todd is making is, depending on how you took the two parts of the data, we called them training and testing, depending on how you took them, could it be interesting if the bottom half is different from the top half? For many purposes, the split is arbitrary, and in that case, they're equivalent. They're the same data often. Right? Often literally. It's often actually a symmetric matrix because it was done split half. And so it's a symmetric relation. Distance is symmetric under normal Euclidean circumstances. The distance from A to B is the same as the distance. That doesn't have to be true if the two halves are not symmetric or if they're not arbitrary. So there can be cases where they're non-symmetric matrices, but in the vanilla case. But so for this case, I drew it as 0, 1. That is a qualitative prediction that says I predict these values will be higher than those values, and that can do a better or worse job of fitting to data. That's the simplest case to draw. But it is absolutely an interesting, good idea to go from binary predictions to quantitative predictions, and that is the next move once we get this move clear. Yeah, totally. One edit that I'm going to make, this is a notational variant. Some people represent these with bigger numbers means more similarity, closer together. Some people represent them as bigger numbers is distance, further apart. When your higher numbers are similarity, it's a representational similarity matrix. When your higher number is distance, it's a representational dissimilarity matrix. These are obviously reciprocal. But since I drew this with bigger numbers, it means more similar, I'm going to write representational similarity matrix. Some people do it dissimilarity. Some people do it similarity. Worth checking which one it is before you go bananas. Okay. All right, so where we were at was, once your data are in the form of a representational similarity matrix, then in addition to asking about classification, you can ask what is the feature underlying the representation by asking which of these two models looks more like your data, right? Where here again, we have sort of a binary version of that hypothesis, right? Either this middle condition is more similar to the first condition or it's more similar to the second condition, and we're asking which of those two it is. But then somebody, and I think it was Melissa, said, does it have to be binary? Couldn't it be continuous? And of course it could be continuous, right? And so the key thing once you see this format is that any source, any idea you have about similarities can be used to generate a similarity matrix that can be used to compare to your fMRI data. The first spelling out of this idea as a kind of a general procedure for fMRI data analysis was from Kriegskorte. And the example that he gave was a set of visual images. I think there's 75 of them, but it might be 96. Where one of the, so these are all images, where one of the similarity matrices came from single unit recording in monkey IT. So there, what similarity means in this representational similarity matrix is for the population of neurons recorded in monkey IT, the distance, so now each neuron is a dimension, right? You can think of each time you present the image, you record the activity in all the neurons. That gives you where that image is in the space of the neuron's activity. And so for each image, you can compute its position in the multidimensional space of neurons, and then the distance of that position to each other image, right? And then the same thing for the same images, but these data are from human fMRI. The kind of key idea of this proposal is that the single unit recording data comes from 200 neurons, where you're recording their average activity level for a few hundred milliseconds in one experiment done in one place, right, in some lab at some time. The fMRI data is from, say, 1,000 neurons, 1,000 voxels. So we might have 200 neurons, we have 1,000 voxels. It's the average activity over seconds, not over milliseconds. It's in a group of people, not 3 monkeys. Maybe done in a different lab at a different time. And the one general challenge of neuroscience has been, how should we compare activity in neurons in monkeys in 100 milliseconds to blood oxygen in voxels in humans in fMRI? There's so many differences between those two kinds of data. How do you even begin to compare them? And so what Kriegskorte proposed is, well, this is a format of data that once you've translated from your source into this format, then it makes them comparable. And so you could, for example, ask, you can take the whole brain data in humans, right, separate it into different local chunks, and ask which chunk, right, which local region of human cortex in fMRI has a similarity structure most similar to the neurons in monkey IT. Does that make sense as a question? So here, take different local regions in human fMRI, because we record the whole brain simultaneously. In humans, there's no cost to recording the whole brain simultaneously. In monkeys, that would be crazy hard, right? You have to keep putting new electrodes into different places in the brain, cutting different holes in the head. It would be crazy hard to record from the whole brain of a monkey. In humans fMRI, you're doing the whole brain anyway. And so that gives us this natural question. monkey IT, we have one representational similarity matrix. Which local region of human fMRI is it most similar to? And what makes it possible is that the human fMRI data and the monkey single unit recording data can be translated into this common language where they're directly comparable, right? The language of representational similarity matrices. No, right. So the idea here, what I'm trying to depict is that you can create the rows and columns here are images, stimuli, and for each local region, so for each set of voxels, you can generate a full representational similarity matrix that is the relationship of every stimulus to every other stimulus, right? So classification is on the diagonal, right? Can you classify one image from another? The answer to that is yes. But then the full similarity space is all on the off diagonal, right? So one thing to say here, right, once you have a lot of images, probably all of these regions can classify. Right? That is all the way along the ventral stream, including in V1, because these are visual images, they're different from one another, right? You can say which image it is above chance, probably everywhere in the brain. The question is, where in the brain are the off-diagonal similarities similar to the ones that are in IT? And this is exactly where the strength of RDMs comes in, right? This is the key point about the difference between an RDM and a classification analysis, is that all of these brain regions have sufficient information to classify images above chance, but the hope is for different reasons. And those different reasons will show up in different similarity structures of the other images to each other, right? So in a region, again, thinking about it in this logic, in a region where you can classify one image from another because of where the bright patches and where the dark patches are, right, like V1, then the images that are most similar to one another will be the ones that have the most similar bright and dark patches. That still means each image is more similar to itself than it is to the average of all the others. But which of the others it's more similar to will be determined by things like the light and dark patches. Whereas if it's a region that has information about the identity of the object, is it a face or a house, right? Again, each image will be more similar to itself than it is to all of the other ones. But which of the other ones it's more similar to will be defined by whether there's a face or a house in it, not by where the light and the dark patches are. And so this is the key idea of an RDM, is that even though in many different regions you can classify above chance, you can try to diagnose why you can classify above chance by looking at the structure of the similarities in the off diagonals. I mean, a correlation is a specific version of a regression, and I'm sure you could do it as a more generalized version of a regression, right? Any more generalized relationship that asks about the explained variance in one RDM by the other one. I have mostly seen that implemented as a correlation, but there's no reason why you would have to do it that way. This is the label version, right? This version, the competing multiple different versions against each other, yes. But in this version, what we're saying is we're labeling our stimuli in different ways and asking which set of labels does the best job capturing the representational similarity. This version day looks a lot like a regression, right? It's just labels. In this case, it's continuous, right? And so that part of why I went to this example is that obviously monkey IT data, right? This is not about a 1 or a 0, right? What's going to be in all of these off diagonals are all continuous measures of distances, like what I drew up there, where for each example category, you have the distance to all the other categories in the similarity space. And in these examples, part of what I'm trying to highlight for you guys is that, so especially in these kinds of cases, classification is not that informative. You can classify an image compared to many other images for lots and lots of reasons. Those might include the high-level percept of what's in the image. They might include low-level confounds. It could be color, could be motion, could be brightness, could be size. There's many different features that could let you classify an image. And so that could give you this case where many different brain regions would have enough information to classify this image as opposed to all the others above chance. But in those cases, the structure of the similarities, which other images it's more or less similar to, could be diagnostic of various things. So what this example is, it could be diagnostic of which brain region is more similar to monkey IT. But once you've seen these two examples, hopefully you can see that there's a huge set of possibilities for how you generate a representational similarity matrix to compare to your data, right? So this is how you generate the data. Now, what this should open up is the possibility of generating a prediction for your data from almost anything. And so what papers like this ideally might look like is you think of a set of stimuli and their similarities such that there are many different ways they could be similar to each other, right? Some of those might be the ways you're interested in. Some of them might be confounds. And for each of those, you can generate a similarity matrix. In an example, again, a different paper with Amy, we had stimuli that were short verbal paragraphs. And for those short verbal paragraphs, we generated something like 8 different similarity matrices. The 20 by 20 data set. But then we can generate predictions based on a whole different set of features. Some of those features were confounds, right? Like length of the paragraph or complexity of the verb structure, reading time. And some of them were variables of interest, the things that we cared about in those stimuli. But each of those features can generate its own representational similarity matrix. And so that lets you start to test when we could classify these stimuli in this brain region, which of these possible features does the best job of explaining why, in terms of capturing the full similarity space over those stimuli. So what Kriegeskorte envisioned as the application of this idea of representational similarity matrices is that we could then think of some set of stimuli, whatever they are. So this, again, this could be a set of different faces, or a set of different objects, or a set of different movies, or a set of different sentences, where there's many different possible sources of the theoretical RDMs. And this, in a sense, becomes a model selection problem, where you're asking which theory generated the RDM most similar to the data. And in principle, and again, this was the vision Kriegeskorte offered, if you collected a good data set, right, one worth explaining, right? Like imagine you had the similarity matrix in some brain region for a random sample of images on the web or a random sample of movies or something. Then you could have a competition among your current theories, but if you later on came up with a better theory, you could ask whether that theory did a better job of explaining the data. So the data you could collect one good data set and have a progressive process of trying to compete to explain or predict that representational similarity matrix. So that was this original data set with the 96 images he offered as an example of that, where once you have a good high quality measure in each local brain region of the similarities in responses to all those images, now each successive theory of what information that brain region is computing could be used to generate an RDM. And now there's kind of one currency that different theories can compete against, which is how well do they generate a similarity matrix that predicts the true similarity matrix in the data. When even our best theory isn't perfect, that's a clue to look for what are we missing, right? Which parts of the similarity structure are we explaining well? Which parts are we explaining poorly? And maybe that's good grist for thinking what parts of the process that this brain region implements does our theory not yet include. In this vision of progress of cognitive neuroscience, where we get one really good RDM and then we do cumulative generating and testing of different theoretical RDMs, a lot of weight goes on, did you choose a good sample to begin with, right? Was it interesting, the similarity space of the sample that you chose? And I think that's really important. So after lunch, we're going to talk about the difference between encoding and decoding models. And as we get here, you can see the ambition starts to be explaining the space, the representational space of a domain, which means that how we sampled our stimuli and whether it's worth explaining the similarities among them starts to become more and more important. And so if that's the question, how do we know we sampled stimuli such that the similarities among them are important to explain and worth explaining, and sample the space of things worth explaining in this brain region? I think that's exactly right. Once you see this way of thinking about the data, it brings forward or centers the importance of how you generated those data. That's the absolutely right intuition, right? And so in the case of the Kriegskorte data set, right, that exactly happened. The biggest difference in IT in the regions that they encoded is the regions that they recorded is between faces and objects. And so it fits best to parts of the ventral visual stream that distinguish faces from objects. Of course, we already knew that, right? We already knew that there's a big difference between faces and objects. And if that is the huge feature in your representational similarity matrix, which it is, right? There's a square for the faces and a square for the objects. Right? That's going to limit what you're able to see in this relationship in good ways and bad ways. Right? It will focus your attention on the faces versus objects distinction. It will limit your ability to see anything else. Right? And we knew about the faces versus objects distinction. Right? And so these questions about, well, what went into this matrix become really important. Yeah. And what is it representative of? I think that's absolutely right. But just to think about, assuming that you had a good data set, right? How do the different theories compete to explain it? Right? That's the key thing that I'm trying to suggest, is that we can think of these different, and one of the things Kriegskorte pointed out is these can be binary matrices, right? They can be simple predictions of what is identical and what is non-identical. They can be continuous matrices that are generated from quantitative theories, right? How similar are these things? They can be behavioral data. So you can ask people, for example, you can do reaction time or discrimination tests. You can look at how behaviorally similar are they. They can be other modalities of neuroimaging, right? So they can be single unit recording. One thing that Ud Oliva's lab, Sishi, what's his first name? Radek Sishi, right? What he showed you could do with this is relate similarity matrices from MEG data to similarity matrices from fMRI data. So MEG has temporal resolution, fMRI has spatial resolution. So he could ask, when in time in MEG is the similarity matrix similar to where in space in fMRI? That's a really cool use, right? And so in general, once you can get your data into the form of a similarity matrix, that lets us cross species, cross modalities, cross different kinds of resolution, compare a priori theories to behavioral measures in competing to explain variance, right? So that's and the power here is that once you have generated a representational similarity matrix, these measures, right, R or whatever generalized measure of explained variance they are parameter-free fits, right? This is a single measure. And so, again, because this is a huge amount of data measured in each of two ways, you might think in general there's a ton of different ways you could relate those two data sets to each other, which gives a huge parameter space of experimental degrees of freedom, and therefore a lot of uncertainty in any relationship that you find. The advantage of this framework is that once you translate all your data into RSMs, there's one test and it happens one time. And no matter how big your RSM is, there's only one measure that you get out of it, which is the overall fit to the similarities space. So that is a huge advantage of this procedure, is that even though your data are really complicated and your theories are really complicated, in the end, it comes down to a single measure that's done once. Yeah, there's two ways to do this. What I prefer to do for statistics is to say, for each participant, there's the data, right? And so these data, you measure the R value, right? This is my preference, is this per participant. And then statistics are across participants. So you get each participant, fit it to each of your theoretical RDMs, and then convert R to Z so that you can do statistics, and then do statistics across participants. The most interesting thing is comparing the models usually, right? So asking whether one model does a better job than another model, especially if those different models are interestingly competing, right? So saying things like the theory-derived prediction does a better job than another theory and a confound, right? And over and above, for example, behavioral similarity, right? So yeah, for example, if you think that inside the brain, the same stimulus gets transformed into a sequence of different similarity spaces, right? Then one thing that's cool is to show that different brain regions are best fit by different feature spaces. This goes back to the idea I said before, right? You could say in some brain regions, feature spaces based on the configuration of the faces do the best job, whereas in other brain regions, more amodal features about the meaning of the stimulus does a better job. And so that kind of direct comparison that says two theories, two brain regions, theory A fits better in one brain region, theory B fits better in another brain region, that could be awesome, right? And then that better, statistically, you would do across subjects. This you wouldn't correct for multiple comparisons if you're comparing them directly to each other, right? Asking whether one is better than the other. If you had one theoretical RDM and you asked, does it fit the data in any of these 6 regions, better than chance, then you would correct for multiple comparisons. It depends how you do it. If you did, so the way Krieger-Skrota does this is he says, look, model the fit of all the models, compute the fit of all the models, each one time, and pick what fits best, then you're not correcting. The goal of fully explaining this particular RDM is valuable in proportion to how representative this RDM is of the brain region you drew it from. And that is related to how you sampled the stimuli, right? And so the question that you're saying is, well, look, in any limited sample, right, like 96 images out of all the images in the universe, there are going to be confounds. And so perfectly fitting the RDM of that set of 96 images probably will not perfectly generalize to all other future images. And so I definitely agree that once we set as our goal explaining all the variance in the representational space of a brain region, we have to start worrying about the limits and biases of the that we use to sample the full function of that brain region. And again, I think that comes out both here and also in the encoding models that we're coming to, and is totally valid, right? So, and in some ways that was sort of the point I was making above, right? For any time your theory predicts the data that you got, you have to ask whether you overfit the data that you acquired. And that will definitely be a consideration here, right? If we only have 96 images as our representation of the function of IT, once we've perfectly represented those, will that generalize outside? But as soon as you do it that way, it's obvious how to test it, right? When you're comparing not just different within the same brain region, but the same model in different brain regions, then what about the fact that in different brain regions you could have different quality data? And in general, of course, you could imagine many situations in which you could have different quality data, like different subject populations, right, might generate different quality data. And so the question is, what about the fact that this number has to be lower when your data are lower quality than when they are higher quality? That is definitely true, right? You can't fit noisy data as well as you can perfect data. And there's a number of possible strategies for handling that. But here's one of them. If the diagonal of your data set, right, so the replication, in the kind of example I've been drawing above, where the rows and the columns are a replication of the same thing, basically, right? So, for example, in Krieger-Skrota's example, this would be 96 images, and then the same 96 images presented again, right? So then the diagonal is a measure of how replicable the pattern is. Right? If I do an experiment and then an experiment again, the diagonal is an estimate of the distance between the first version of the experiment and the replication of the experiment. And you can think of that as a measure of data quality, right? It's a measure of how replicable the pattern is if you do exactly the same thing again, right? Is that clear to people? And so one thing you can do is use that to set what sometimes is called the noise ceiling, which is you can only explain as much variance in your as is signal, right? You can't explain the noise because it's not replicable. And so we can use the diagonal as an estimate of how replicable the patterns are in this data, full stop, right? The test-retest reliability of the data, and say that's as much as we could possibly explain, right? The similarity of the same condition to itself again. So you can use, there's various ways of using that similarity to compute how much of the variance in your matrix is real signal variance and how much is noise. And then compare your theory to your data as a function of that ceiling. And that, if that ceiling is different across different regions or different populations, then that will let you say, okay, even though in the kids the data were just noisier than the adults, once you map the matrices by this measure of their replicability, then we can ask, do different theories fit the data to different degrees? Or if frontal regions compared to occipital regions are more noisy, just less replicable, we can say, okay, but relative to the replicable stuff, how do the theories fit? So that is something Kriegskorte encourages, and many people do. There's different ways of implementing it. But again, the key idea here is if the diagonal is a measure of replicability, test-retest reliability, then you can use that in a way to compute how much variable data is explainable by a theory because replicable. If you think that everything you should be able to explain should repeat in a replication of the same experiment. Everybody is doing this stuff now. Yeah. So he was early to the game, but this is very widespread now. So Oliva and Radek Sishi have done the stuff relating it to MEG, but essentially every group. So this is a question about essentially about permutation as a way of doing statistics. Certainly, you can't just say, is the average of these values greater than 0, right? For lots of reasons, including that they are non-independent measurements. And so, right, there are questions about the best ways to do statistics over representational similarity matrices if you're not doing theory comparison. And label shuffling, which is the question you were asking, right? Where so you take the same data set but shuffle the labels in the rows compared to the columns and iterate that as a way of generating the null hypothesis is certainly one way to try to estimate empirically what would null data look like sampled in this way.